Hello. Hello. Good morning.
Yes, ma'am. Good morning.
Okay, thank you. And I think you have read or have notes of the previous classes. Have you got those notes, Sipul? Yes. Hello, Sipul.
Yes,
Have you
ma'am.
done your notes?
No,
Okay,
ma'am.
thank
No.
you. Also, I think you should. Oh, you didn't. Okay. Hello, Gayatri. Have you done your notes of the previous classes? Hello, and write your roll numbers in your chat boxes, students. Hello, Gayatri, Sripun.
Yes, ma'am.
Write your roll number. Okay. Thank you. Gayatri, you are from
Yes, ma'am.
which place? Yes, Gayatri, you are from which place?
Bhubaneswar.
Bhubaneswar. And Sachin, you are from?
Ma'am, I am from Dhenkanal.
Mm. And what type of careers you are doing now? Or service holder, Sipul?
No, ma'am. I'm still studying. I have just completed my DLD course city.
Oh, yes. Yes. And Gayatri, what are you doing now?
Yes, ma'am. I'm studying and training Noji horticulture.
Horticulture. Horticulture in OUT?
No, ma'am. Chase Central Horticulture Experimentation.
Mm-hmm. Very nice. Rupali, you write your roll number. Good morning. Students, you write your roll numbers. Anuradha, you didn't write your roll number. Anuradha, Aliva, write your roll numbers.
Yes, ma'am.
Students, you all are requested when you are joining, just write your roll numbers in chat box. Okay.
मैम ମୁଁ ସେଇଟା କରିପାରୁନି ରୋଲ ନମ୍ବର କୋଉ ଚାର୍ଟରେ ଲେଖିବି
chat box ର ଦେଖ chat box ର ତୁମର chat box ଥିବ ଯୋଉ ତିନିଟା ଡଟ୍ ଥିବ ଦେଖ ମେସେଜ ର ତ ମୋବାଇଲ ରେ କରୁଛ ନା ଏଥର କରୁଛ ଲାପଟପ ରେ
ମୋବାଇଲରେ ମ୍ୟାଡାମ
ମୋବାଇଲ ରେ ତୁମେ ଦେଖ ତାର ଗୋଟେ ଅଛି ତୁମେ ଯୋଉ 3 ଡଟ୍ସ ଥିବ ତଳେ ହେଲା ସ୍କ୍ରିନ ର ତିନିଟା ଡଟ୍ ରେ ଦେଖ ସେଟା କ୍ଲିକ କର ଆସିଯିବ ମେସେଜେସ୍ ଓକେ ମେସେଜ ଖୋଲିଲା ପରେ ତୁମେ ସେଠି ତୁମର ଥିବ ବ୍ଲିଙ୍କର ଥିବ ତୁମେ ସେଠି ଲେଖିଦବ ତୁମ ରୋଲ ନମ୍ବର ଖୋଲିଲ
ହଁ ମ୍ୟାମ
ପାଇଲ
ଜାଣିପାରୁନି କୋଉଥିରେ ଲେଖା
ତୁମ
ହେଇଛି
ମୋବାଇଲ ଯୋଉ ତୁମେ ଅନ୍ ସ୍କ୍ରିନ୍ ଅଛ ତୁମେ ଦେଖ ତଳେ ରାଇଟ ସାଇଡ ଏକ୍ସଟ୍ରିମ ରାଇଟ ସାଇଡ ରେ ତିନିଟା ଡଟ୍ ଅଛି କିମ୍ବା ଉପରେ ବି ଥାଏ ତିନିଟା ଡଟ୍ସ ଦେଖିଲ
ଉପରେ ଅଛି ହଁ
ଉପରେ ତିନିଟା ଡଟ୍ ରେ କ୍ଲିକ କର
ହଁ
କଲ
ମ୍ୟାଡାମ ହଁ
ଦେଖିଲ ମେସେଜ ର ଗୋଟେ ଆସୁଛି 5-6ଟା ଖଣ୍ଡେ ଥିବ ଇନକଲ ମେସେଜେସ୍ ଲେଖା ହେଲା ଅଛି
ନାଇଁ ମ୍ୟାଡାମ ସୁଇଚ କ୍ୟାମେରା
ହୁଁ
ଟର୍ନ ଅନ୍ କ୍ୟାପସନ୍ସ ପ୍ରେଜେଣ୍ଟ
ହୁଁ
ସ୍କ୍ରିନ୍
ହୁଁ
ରିପୋର୍ଟ ଏ ପ୍ରୋବ୍ଲେମ
ହୁଁ
ରିପୋର୍ଟ ଆପକସ୍ ନା କଣ ଏଟା ଆବ୍ୟୁଜ ହେଲ୍ପ କ୍ୟାନ୍ସଲ
ଓ ଆସୁନି ନା ତୁମ ନାଁ କଣ କହିଲ
ଅଲିଭା ଜେନା
ଅଲିଭା ଜେନା ଏଇଟା କଣ ଓଲ୍ଡ ଭର୍ସନ ଟା ନା କଣ ଥିବ ଦେଖ ତୁମ କୋଉ ସାଙ୍ଗ ମାନଙ୍କୁ ହେଲେ ପଚାର ଟିକେ
ଅନ୍ୟମାନେ ଯୋଉଟା ଲେଖୁଛନ୍ତି ସେଟା ଗୋଟେ 1 2 ଆସୁଥିବ ଯୋଉ ଏନରୋଲମେଣ୍ଟ ଲେଖୁଛନ୍ତି ସେଟା 1 2 ଆସୁଥିବ ସେଇ ଚାଟ ବକ୍ସ ରେ ସେଇଟା କ୍ଲିକ କରନ୍ତୁ ତାପରେ ସେଇଟା ସମସ୍ତଙ୍କର ଆସିବ ଏବେ ରିସେଣ୍ଟଲି ଯୋଉମାନେ ଲେଖିଛନ୍ତି ସେଇଟା 1 2 ଆସୁଥିବ ମେସେଜ ଆସିଲେ ଚାଟ ବକ୍ସ ରେ ସେଇଟା କ୍ଲିକ କରନ୍ତୁ ସେଇଟା ଆସିଯିବ ମେସେଜ ଟାଇପ କରିବା ପାଇଁ
ଅଲିଭା ଜାଣିପାରିଲ
ଚେଷ୍ଟା କରୁଛି ମ୍ୟାଡାମ
ହଁ କର ତାହେଲେ ଲାଷ୍ଟ କ୍ଲାସ ରେ ବି ତମେ ଲେଖି ପାରିନଥିଲ ନା ରୋଲ
ନା
ନମ୍ବର
ମ୍ୟାଡାମ
ଓହୋ
ନା
ତମେ ସାଙ୍ଗମାନଙ୍କୁ ତା ଭିତରେ ଟିକେ ପଚାରିଦବ କେତେ କ୍ଲାसेସ୍ ମିସ୍ କରୁଛ ତମର ଆଟେଣ୍ଡାନ୍ସ ଟା ଇଏ କରି ହଉନି ନା ହେଲା କି ଅଲିଭା ହେଲା
ହଁ ମ୍ୟାଡାମ ହେଲା
ହେଲା ଓ you got it thank you okay we should go to the classes okay I'm starting my screen sharing okay students you all have written your roll numbers in chat box
Yes, ma'am.
okay
Yes,
thank you
ma'am.
okay thank you
ମ୍ୟାଡାମ କରିଥିଲି
ହଁ ହଁ ମୁଁ ବି ଦେଖିଲି ତମ ରୋଲ ନମ୍ବରଟା ଅଛି okay is it visible to all of you
Yes, ma'am.
Okay, unit say again. Yes, extranuclear inheritance.
Inheritance.
Yes. Yes, so that means it is outside the nuclear. No, extra nuclear means it is out of the nucleus, because we all are accustomed to that inheritance that is related to the DNA and the gene. When it is called extra nuclear means extra genes are found in cell organelles except the nucleus, out of the nucleus, such as mitochondria and chloro mitochondria and chloroplast. Am I audible to everyone?
Yes,
Hello?
ma'am.
Okay,
Yes, ma'am.
thanks. Yes, thank you. So, via the extranuclear genes, the characters are inherited to next generation. That means through the mitochondrial DNA and the chloroplast DNA. So, it's known as cytoplasmic inheritance. Naturally, it should be called as cytoplasmic inheritance because it is out of the nucleus, or extra chromosomal inheritance. Okay, and it was actually discovered by the scientist C. Corvinus during the 1909 and E. Bohr 1909 and to 1910 described crossings for which the Mendelian laws didn't apply, because the Mendelian laws are restricted to the genes or chromosomal genes. See here in this screen what happened in step 1 and step 2. So Corens perform the reciprocal process between G and variegated mirabilis plants, and I think you all are well known to mirabilis plant. Have you seen mirabilis plant, students?
Ma'am,
Mirabilis plant. Mirabilis plant. Okay, I am joining someone. Okay, just a minute, students. Okay, Mirabilis plant, have you seen? Anybody? What is Mirabilis plant? It's the scientific name. Or 4 o'clock plant. Is there anybody who know Mirabilis plant? Oh, okay, Mirabilis plant, just to search your Google, that is the ଓଡ଼ିଆରେ ତାକୁ କହନ୍ତି ରଙ୍ଗଣୀ ଫୁଲ, and that is not available nowadays in our garden. Okay, in Mirabilis plants, so using sectors, because ଏଇ ଯୋଉ Mirabilis plant ଆମର ରହୁଛି ନା, ସେ Mirabilis plant ରେ ଆମେ ଯଦି red ଆଉ white ର ତାକୁ ଆମେ hybridize କରନ୍ତି, ତାର ଗୋଟେ ଆମର ଆସେ ଦିଟା ଯୋଉଟା କୋଡୋମିନାନ୍ସ ଦେଖାଏ. ଓକେ, ତାର patches flower ସବୁ ଆସେ. ସେଟାକୁ ଟିକେ just to observe that. So in mirabilis plants, using white sector on the variegated plants, ସେଥିରେ ଆମର ଆସିବ gametes. ଦେଖ, just see here in this left hand screen. So green plant, variegated plant, and the options will be green. Okay, and in case of the second experiment, what happens? From each cross, the offsprings show the same phenotype as the female parent. If female parent is green color, so all they are showing that the characteristics of the female plant. And here, in case two types of pollen, white or green pollens are found, and that are used to fertilize the flowers. And here, branches with white leaves gave only white leaf progeny, and the green leaves that give only green progeny. Just a minute, someone is being admitted. Okay, students who are coming during the classes, please write your roll numbers in your chat blocks. Again, I am requesting dear students, write your roll numbers in chat box. Okay, and what happens here? White leaves, they give the white progeny, and the plants with green leaves give green progeny, and branches with variegated leaves, mean different leaves, those get green, white, and variegated progeny. And here, during this observation of the phenotype, means the phenotype doesn't depend on the nuclear genotype. Okay, so pollen here the pollens are not involved, and that must be dependent on cytoplasm of female organs in the flower. And cytoplasmic inheritance, during this cytoplasmic inheritance or maternal inheritance, because that is mostly adopted or that is inherited from the female or the egg. The cytoplasm organelles such as mitochondria and chloroplast are inherited with the egg cytoplasm from the maternal parent. The example is cell coiling in snail and variegated leaf color in the 4 o'clock plant or mirabilis plant, that is known as the 4 o'clock plant. And here this important point you should remember that mostly the cytoplasm um, inheritance that is associated with the egg cytoplasm. And see here how the cell coiling in snails is occurring in this slide. That one is dextral variety, that is dominant variety, and the sinistral variety uh, is the recessive one. In generation 1, what happens? The egg, T is the egg and 3D is the sperm. When we are crossing both the varieties, we are getting, see in this picture what type of varieties you are getting. So from generation 1, uh, see, under cell fertilization, when cell fertilization takes place, in we are getting that in generation 2 and in generation then we should okay. Hello, who come late? Okay, write your roll number in chat box, please. Students, write your roll number. And what happens in generation 3? Just see what type of offsprings are formed. In generation 2, we are getting dextral type. Naturally, the dominant variety would conceal the recessive variety. So, in first, second generation, you are getting all the dextral variety, and the when we are getting self crushing among themselves, we are getting all the in generation 3, we are getting all dextral varieties, and here also um, all the progenies are all dextral varieties. But in generation 4th generation, you see the hybridization among themselves that gives 50% dextral, 50% sinistral varieties. Okay, and in case of the infectious inheritance, if naturally there will be some infectious cases in these cases also, the extrachromosomal inheritance. So the cytoplasm of animal and plant cells is very complex. In addition to the normal cellular components, numerous parasites can infect the cytoplasm and replicate there also. Also, example is sigma virus in Drosophila, the most accepted example. That is, you should remember that sigma virus. Have you heard about sigma virus, students? Is there anybody who have listened about sigma virus, students? Hello.
No, ma'am.
Okay. Okay, oh, that is present, that is infectious virus in Drosophila. Okay, should I ask, all have written your roll numbers in chat box, dear students?
Yes.
Okay, thank you. Okay, so this sigma virus in Drosophila, that is the most important example. And these parasites include the bacteria and viruses. So this is termed as infectious inheritance. So when a bacteria or virus those infect the cytoplasm, so that is known as the infectious inheritance. Okay, just a minute, students. Someone is joining. Yes. Hello, Aravindo. Write your roll number in chat box, dear. Hello. Hello. Am I audible to you, Aravindo? Hello, Aravindo?
Yes, ma'am.
Please write your roll number in chat box. Okay, then come to the human chromosomes. And how many pairs of human chromosomes are there?
23 pair, ma'am.
Okay, and see here how the karyotype or in this figure 22 pairs of autosomes are there, and one pair of sex chromosomes are there. Okay, see here the number of pairing 22 up to 22 pairs are known as the what autosomes. Am I right? And in case of the humans, see how the karyotype of sex chromosomes X is larger than the Y chromosomes, and here this X chromosome is similar to all the chromosomes except that Y chromosome. It is the smallest one in the karyotype of human beings. Okay, and in case of the females, we are getting double X, and in case of the males, we are getting one X chromosome and another Y, that is from the father. And in case of karyotyping, how we are karyotyping or the arranging the chromosomes. So karyotype means it's the organized profile. We are arranging. We should arrange as per their thickness, length. Okay. So this arrangement of the chromosomes is known, or profiling of the chromosomes of a person is known as the karyotype. So here chromosomes are arranged and numbered by size from largest to smallest as per their size. This helps scientists quickly identify the chromosomal alterations that may result in genetic disorder. Okay, and the process in a laboratory process, see here that some tissues are collected from the persons, and chemicals are added to it for the culture in growth medium, and after 2 to 3 days of incubation, again chemical is added that restrict the mitosis process in metaphase, and as we have to take the photography or karyotype or profiling. So mitosis must be stopped by adding a chemical, and that is transferred to the tissue culture is transferred to a test tube and again centrifuge to concentrate in the layers. Okay, and here uh, again after the transfer they are added by a fixative for the staining, and those fixative helps in studying under the microscope. Okay, then after the identification under the microscope, we are getting this type of karyotype. Okay, see here in the figure. So karyotype arrangement or karyotyping by the laboratory process. Okay, Then one type of chromosome banding is also there for the study of the human chromosomes. These are the Q banding, G banding, and C banding. Okay, in Q banding, chromosomes stained with a fluorescent dye such as guanine, quinine, and in G banding, so you have to take these chemicals in G banding. What happens? That those chromosomes are stained by a giemsa stain. Okay, after digesting the chromosomes with the trypsin. So in G banding, trypsin is one. What is trypsin? What that is one type of enzyme, digestive enzyme found in our digestive system. Okay, in small intestine, that is extracted and that is added for the digestion of the tissue region. Okay, in C banding, the chromosomes are treated with acid and base, then stained with again giemsa stain. So for Q banding, giemsa stain is used, and for C banding also giemsa stain is in three varieties. Giemsa stain is using, but here in G banding the trypsin enzyme is used, but in C banding that is the acid is used. Okay, making how we are making a karyotype, just we are arranging from a larger to smaller ones by its size. So first the total number of chromosomes is given, followed by a comma and the sex chromosomes confusion it is get separately. Okay, and in next slide, preparing a karyotype. How? Or during the metaphase cell, actually the karyotype is carried out during the particular stage, that is metaphase. Okay, we have given a chemical during that chemical process in laboratory to stop the metaphase stage. Okay, so during metaphase cell the preparation of karyotype is very essential, and a population of dividing cells could be used. Blood culture, skin fibroblasts, bone marrow cells. So these types of tissues are used, and also lymphocytes induced to proliferate. Okay, and many protocols, but a standard series of steps in is used in the laboratory process, and these are the karyotype protocol. Okay, so see here blood and heparin to prevent, because heparin is used to prevent the clotting of the blood, and centrifugation during that laboratory process we have already discussed that centrifugation is must should be carried out. So these are the blood plus heparin to prevent the coagulation. Centrifugation through dense medium, it is also needed, and cultured for 3 to 4 days. We have already in that previous slide where I have discussed you in during the laboratory processes. Okay, and in the fourth stage, let's see here. Add. Okay, just a minute, children. Hello, Suchismita. Hello, Suchismita. Okay, come to after the protocol points. Just see here. Symmetric and asymmetric karyotypes. There are two types of karyotypes, that is in asymmetric and symmetric. First, you see what happens in symmetric. This symmetric karyotype is lesser, so lesser difference between smaller and larger chromosomes in a set. Have more metacentric chromosomes, have no relatively advanced picture, but in case of the asymmetric karyotype, that shows larger difference. Here lesser difference in symmetric, here larger difference in symmetric. Have more acrocentric chromosomes, here in symmetric that is more metacentric, and here have relatively advanced feature in asymmetric karyotype, and here no relatively advanced features. Okay, Suchismita, hello? Suchismita,
Yes, ma'am.
have you written your roll number?
Yes, ma'am.
Hello?
Yes, ma'am. I have written my roll number.
Okay, thank you. And then come to the replicated that telocentric, acrocentric, submetacentric, and metacentric chromosomes. Here, the figures you have already accustomed with the figures during a class 2 courses. Okay, so in telocentric, the centrosome present at the far end of the chromatid arm. In acrocentric, that is a very small arm is present, so it is far end of but having a small chromatid arm, and in metacentric that is present in the center, and it is in case of the submetacentric it is just far from the centrally present centromere. And come to the banding technique of the chromosomes. In banding technique of the chromosomes, we have to denature the protein. So weak trypsin or urea or protease, they may be added to denature the protein. Okay, then treated with gymsastain. This is also a graphical figure of that um, our laboratory processes. Okay, then here what happened when gymsastain is added, here interaction of the DNA with thiazine and uracil component of the stain brightens the sulfur rich regions. You know, sulfur rich regions are all present in your DNA configuration. Okay, and here in addition of the gymsastain, methylene azure, methylene violet, methylene blue, and eosin, they are also now added for the banding of the chromosomes. Okay, and in bending chromosomes techniques, what happens? The first the chromosomes are air dried and treated with 5% tri chloroacetic acid in a regulated temperature, that is 95 degrees centigrade for 30 minutes only. Then their chromosomes are treated with HCl, that is one acid, and at the 60-degree temperature for 30 minutes. Okay, banding pattern. Then these banding patterns are observed. Okay. Uh, Next, in C bending techniques, what happens? The chromosomes treated with alkali solutions. Okay. Here DNA is get denatured. Then washing with sodium citrate at 60 degrees centigrade for 30 minutes. And here this washing is also repetitive process. Okay. Then staining with gymsastain, and after that we are getting the bending patterns of the chromosomes. Then come to the next unit, structural abnormalities in chromosomes and their effects. Here, what type of structural abnormalities may be there? So these are group of problems that is caused by the structural abnormalities. So what type of structural abnormalities are there? So in this point, some abnormalities are balanced and some are unbalanced. In balanced cases where there is no loss or gain of the chromosomal material, that is one type of abnormality is observed. No loss of the chromosomal material. But in unbalanced, here the individuals has more or less chromosomal material than usual. What should be present in a chromosomal material? So here some extra material may be present, or some material may be okay. So here, so that is naturally that will be unbalanced. And here what happens, uh, These abnormalities may occur in germ cells or somatic cells. So in both type of cells, we are getting the abnormalities or abnormal forms. What happens in these abnormal forms? Those that happen in germline or germ cells cause difficulties in egg or sperm development and normal development of a zygote. When that occurs in germ cells or germ cytoplasm, naturally the sperm or egg development that may be anomalous. This can be transferred to next generation, and those happen in somatic cells other than the sex chromosomes or the 22 pairs, so that may cause cancer but are not transferred to the next generation. So that is restricted to one generation. Okay. So in somatic cases, that is not inheritable, but in case of the germplasm or sex cells, so they are inheritable to the next generation. Okay. Okay, students. Then come to the structural abnormalities. How, in where, or the position of the chromosome. So translocation means ratio. One is translocation, second one is deletion or micro deletion. Some part may be deleted. Okay, and third one is the duplication. Fourth one is inversion. Fifth one is insertion. Insertion means addition. Okay, so translocation means it is the in next slide we will discuss about this translocation. And deletion totally may be deleted, some portion may be deleted, or a micro deletion may occur. Duplication means it will be duplicated, and inversion means inversion, the pericentric inversion or paracentric, that will be coiled or a disfigure of the chromosomal, because these are present in a linear fashion, so there may be a inversion. And insertion means addition, new chromosome material or gene material, they may be added. And in sixth point, ring chromosome. Next is the isochromosome, and the eighth one is the chromosomal breakage or instability. That is one example of the Fanconi anemia and blood syndrome. So these 8 type of structural abnormalities, just see here in this slide. Or translocation means, see here, some portions they may be translocated, and deletion means totally deleted. Here, see here, one inversion means the part may be inverted. Inverted one, their arrow mark. Just see here in this inversion, what happened? That is totally inverted. Inversion means inverted. Okay. And in isochromosomes, what happens? See here, totally the centromere or the portion of the chromosomes, they are also rearranged. Okay. And insertion means, just see, in case of the insertion means new addition of some portion. And derivative chromosomes means one pink size chromosome is newly derived from the blue one. Just see here in derivative chromosomes. And in the ring chromosome, the chromosome may be a ring size due to some effects. That may be chemical, that may be environmental. Okay, so these are the structural abnormalities we are getting during our lifetime. Okay, in what happens in translocation? See here, a fragment of a chromosome is moved or translocated. Okay, transform ଯେମିତି ଆମେ କହୁଛନ୍ତି. Okay, so translocated location ଟା ତାର ପୁରା transfer ହେଇଯିବ from one chromosome to another joins a non-homologous chromosome, and that is transferred from one chromosome, and again that material may be joined to another chromosome that is non-homologous. Just we are telling that what is homologous and what is non-homologous. Homologous chromosome means a pair of chromosomes. They are derived from paternal, one from father and another from mother, and they are similar in size and their origin. Okay. From this pair, they are totally deleted and translocated to a non-homologous pair. Okay, who is coming, student? Hello? Hello, who are coming during the classes, write your roll numbers in your chat box, please. Okay, so here in translocation position, so they join or they get fragmented from the homologous pair and they join to another pair. Might be the second pair may be joined to the third or 13th pair. Okay, these are because the second pair is non-homologous to the 13th pair. Okay, so here this type is also two types. One is reciprocal and another is Robertsonian translocation. Okay, and in deletion, I have already told you that in deletion process, totally the portion of one chromosome is lost. In translocation, that is fragmented from one chromosome and added to the new chromosome or non-homologous chromosomes. But here, the portion from one chromosome that is totally lost. Okay, and that may be terminal or interstitial or naturally or normal chromosomes, terminal deletion and interstitial deletions may be occur. Okay, and in case of this deleted process or deletion, so some segments most are lethal, and here these are causes very serious degenerates in a human being. Okay, deletion because that is totally deleted. So some gene expression might be deleted from our blueprint of life during our life processes. So these are very serious disorders in a human beings. And the third one is the duplication. In duplication process, initial duplication of a single copy. See here in this figure, what happens in a duplicated process. Just see what happened during genetic exchange sites. So A, B, C, the three portions of a chromosome, two arms A, B, C, and here in this positions A dash, B dash, and C dash. What happened during the duplication process? See the position of the deleted chromosome, and B is deleted here. See from the interspace between the A and B, and that place is deleted and again joined to the second arm. Okay, so that is duplicated in the second segment of that chromosomal arm. Okay, and in inversion, what happens? The linear stage of DNA is reversed with the chromosome. How it is reversed? See here the pattern of the portions. Just see here in A, B, C, D, E, F, G, H, I, which I have in the slide G, H, I, and this G, H, I that is reverse I, H, G. Okay, so it is known as the inversion process. So their configuration just that changes. G, H, I that is inverted into I, H, G. So this also occur in case of the chromosomal abnormalities. And in case of the insertion, before insertion, just see here chromosome number 4 area being inserted. How the chromosome portion, a newly added chromosome, how it is inserted in the chromosomal arm. See here, after inversion, what happens? This new chromosome 20 is inserted in the chromosome number 4. Okay, so this is a totally new fragment of a different chromosome that is added to a different homologous chromosome. Okay, and in case of the ring chromosome, what happens in ring chromosome? A ring chromosome is a chromosome which or whose arms have been fused together to form a ring, just like a ring form, a linear form that would be to a ring form. Okay, and in case this ring form, so ring chromosome may form in cells following genetic damage by mutagens like radiation. Mutagen 1 is radiation, and that radiation causes the ring form of the uh, chromosome. Okay, and in isochromosome, what happens? Loss of one arm and duplication of the other. One arm is totally lost. Here in the left side, that is crossed, that portion is totally lost, and the remaining part of the chromosome, that is duplicated again. Okay, so this is the example of the isochromosome. Then come to the unit 10, that is numerical abnormalities. The types or the how many numbers of chromosomes or chromosomal abnormalities we are getting and what are their effects. Then here normal female, we know normal female have 46 pairs with double X, and normal male has 46 numbers with XY. Okay, but in abnormalities, what happens in case of the numerical abnormality means maybe added 47, maybe deleted 45. Okay, and in the sex chromosomes, double X, in place of the double X you may get triple X. You may get in case of the XY, in case of the males we may get double XY. Okay, so these are the numerical or the numbering abnormalities. So any karyotype where the number of chromosomes present in the single cell differs from the normal form or the 46 number ରୁ ଅଧିକ ହେଲେ ଆମେ କହିବା କିମ୍ବା କମ ହେଲେ, that is the numerical abnormalities. And what are their effects in the during the life period of a person? So chromosomal, in case of these chromosome abnormalities, they may reflect either an incorrect number or a structural abnormality. Okay, and in case of the numerical abnormalities, what happens? Most irregular chromosome numbers are result of non-destruction or a failure of meiosis in the production of sperm or ova. Okay, and in that case, about 20% of all human conceptions carry a chromosomal abnormalities. Just a minute, I am joining another one. Yes, Deepak. Hello. Hello, Deepak. Write your roll number in chat box. Hello. Okay. So in most irregular forms, 20% of human conceptions during the conceive of a baby. So 20% we are getting this type of abnormalities, and generally in 1,006% or 6 newborn babies are affected. And these chromosomal abnormalities may be spontaneous or in a defective newborn case or spontaneous abortion. So these abnormalities may also lead spontaneous abnormalities or spontaneous abortion, or if that case persists, so that may produce a defective child. Okay, in numerical abnormalities, we are getting trisomy, monosomy, triploidy or tetraploidy and others. In trisomy, what happens? The 16 number, 13 pair, in the these are the chromosomal uh, numbers. Okay, in karyotype. So in trisomy 16, 13, 18, 20, we are getting triple X or XSY or X double Y. So these are the percentage we are getting in these cases. So the trisomy, monosomy, so the percentage of the spontaneous abortion are occurring in this percentage. Okay. Then types of numerical chromosome abnormalities. In this case, what happens? Numerical abnormalities, the polyploidy and euploidy. In polyploidy cases, we are getting triploidy or 3n number or tetraploid 4n number. And in case of the aneuploidy, monosomy and trisomy. And in case of the types of numerical chromosome abnormalities, what happen in case aneuploidy, that occur in one or more extra missing chromosome, leading to an unbalanced chromosome complement, and that gives the non-disjunction pair. What happens? See here, in metaphase 1 and anaphase 1, see the pattern of the non-disjunction matter. And next slide, we are getting in case of the monosomy, it is a form of aneuploidy, and it is characterized by the presence of only one chromosome from the homologous pairs. Okay. And in case of this one symptom or a disease is known as the Turner syndrome. Here, this syndrome is giving the full moon or full monosomy that is seen in the living individuals, and one chromosome means genotype must be, because monosomy means single pair, so only the X chromosome here present, and their genotype representation would be X0, and karyotype will be 45 because Y is totally deleted here. Okay. Uh, in case of this X0, and the karyotype is 45 S0 type, and only affects this case is mainly occurring females only. Okay. And in case of the partial monosomy, that was the full monosomy. In case of the partial monosomy, what happens? Most partial monosomy is caused by deletion on chromosomes. Some portion will be deleted. Okay, and the example is Chat syndrome or the George syndrome. Okay? And in Chat syndrome, that is the deletion, a portion of the chromosome, monosomal pair, say one, a portion will be deleted. Okay, and symptoms will be, that is difficulty in swallowing, a distinctive cry, poor growth. Okay, uh, and in numerical chromosome and variety numerical chromosome polyploidy cases, this polyploidy, in these polyploidy cases, refers to number of complete genomic content in a single cell, and this gives the uh, digeny versus diantry. And here triploidy cases means 69 chromosomes may occur, or tetraploidy means 92 chromosomes may occur. So that comes in polyploid uh, cases of the abnormalities. And in case of the non-disjunction, what happens? The failure of the chromosome pair to separate properly during meiosis or mitosis, because the chromosomes cross during crossing over, and in meiosis one. Okay, so here the complete failure of the separation of the chromosomes occur. Okay, and one point is there, that is a failure of homologous chromosome to separate in meiosis one, and another one is failure of sister chromatids to separate during meiosis 2, because meiosis 2, that is a complete the repetition of the mitosis and division. And here what happens in maternal increase when the mother is more aged. So in case of a when she is going to be a mother or to conceive, okay, in case of the age old females, this is more example. The non-disjunction chromosomal matter that occur during age old mother when she is getting um, conception. Okay, and another one is disease of this Down syndrome. Okay, in Down syndrome, what happens? A trisomy occur at the chromosomal pair 21, and this disease is known as the abnormalities addition of one part or extra chromosomal part to the 21st number of chromosome, and it is named after the scientist John Down. Okay, he was a doctor. Okay, and its epidemiology, just you see here, in epidemiology the incidence is 1 in 800 to 1000 birth. And in Down syndrome, these are the symptoms. You may get small or low ears, short stature or hypotonia, very impaired intellectual. You might have observed so many cases uh, in our society. So they have a very less cognitive ability. Cognitive means intelligent ability to rethought, to recap their memories, and a very impaired intellectual person, intelligence, and the heartbeat is very low, small head, upward slanting eyes, and flattened facial features also. So they have also so many treatments are there. Uh, so they can be treated through so many medicines and counseling and support. Okay, and some facial plastic surgeries are also available now in 21st century. So this type of syndrome can be uh, treated. And in case this Down syndrome, what happens? Trichomy at the 21st pair, see here. So this is can be observed in a genotype configuration. Just you see here. So these are the 21st trichomy in 21st pair, and that occurs 95% of Down syndrome cases, or 88% occurs in maternal a non-distinction, while a mother is more aged and conceiving an offspring. Okay, and in trichomy occur when in 13th pair, that is known as the Patau's syndrome. Okay, time is very limited. Just I am skipping your slides. Okay, when the abnormality occur in 13th pair, that is known as the Patau syndrome. And in case of this abnormalities, that is another one is Turner syndrome. In Turner syndrome, what happens? The configuration, genetic configuration A is X0, and when triple X syndrome occur, it is known as the also trisomy X. Here, the three X's are present, and the females with the trisomy X usually don't present any significant symptom, because two of the three X chromosomes are inactivated by the non-coding RNA. Okay, so here the problems, behavioral problems will be poor coordination or mental retardation or just infertility cases. So trisomy, in case of the trisomy, it is not observed physically more. Okay, they may be retardation or infertility, mental retardation or infertility cases. And another one is Klinefelter syndrome. That is also this Klinefelter syndrome. Extra chromosomal, just see here, another X chromosomes is added to a male person, that is mostly present, uh, uh, actually found in case of the uh, persons affected, how they are mainly in case of the males, and they do exhibit in some female secondary sexual characters. Actually, males are affected, but they are producing the secondary sexual character, like the development of the breast, okay, and some pubic hairs. And in case of one example is one the portion of 1000 male births. From 1000 males, one per case may be uh, observed in this type of syndrome. And in case of the XYY, where Y is extra added, that is XYY syndrome, that is aneuploidy or a Y chromosome is inherited an additional chromosomal set. So it is also found in one in case of 1000 or one in case of the 2000 male birth. And another one is Edward syndrome or trisomy number 18. It is associated with trisomy number 18, and here the kidney malfunction, heart defects, so many problems may be there, and mental retardation. Here, see here, physical malformation in Edwards syndrome. Okay, and in case of the nature and structure of the genetic material, this genetic in case of the genetic material symptoms, the characters transfer from parent to offspring, and that is mostly associated with the mitochondrial nucleus and cytoplasmic material. And here properties in genetic material syndromes, what happens here, replicated high fidelity, high degree of precision, and high variable information for gene function. And conflict over the genetic material, protein or DNA or RNA materials, they are mostly regarded as the genetic materials. But what happens in case of these problems, and how we could identify these genetic materials by the experimental process from nuclein by mixture, and there was a hunt to know the genetic material experiments. Okay. Dear students, should I continue for more 5 minutes? Hello, students.
Yes, ma'am.
Okay, thank you, thank you. During this experiment, actually, the three scientists, that is Avery, MacLeod, and McCarthy, they have experimented this genetic material experiments, and by the experiment, during their experiment, they take the virulent forms, virulent forms, and live strains, they are injected to uh, mice, and when they are injected to mice, all mice survived. Live strains, when the third dose were injected, all mice are died. And the heat killed IAS injected, when all mice are survived. Okay, and heat killed IAS, and when with added with second IR, they were injected, all mice died. So these are the experiment processes. And next slide, just I'm skipping these experiments. You have to collect. And in the very MacLeod and Curtis experiment, these IIR and heat-killed IIS cells were plated on colony, and T2 hour gave smooth colonies, and all are survived. From the experiments carried out, that DNA, the actual genetic material. They conclude that DNA is a actual genetic material, and these are the experiments of the Hussey and Chase, that how the DNA was universally accepted and with a bacteriophage. How bacteriophage DNA, that is the genetic material. They concluded that, and in case of the E. coli, in other experiment, they cultured S35 phages with E. coli for 10 minutes, and here it is known as the ghost, that outside considered S35 while progeny was normal. Okay, and in our as RNA is the genetic material, so we know that Frankel and Cornet, they independently done an experiment, and they regarded as RNA is also a genetic material. Okay, and in a genetic material, phosphoric acid, pentosugal, those are, and organic nitrogenous bases. These are the three components. And in nitrogenous bases, adenine, guanine, cytosine, and thymine. Okay, and in case of the, I'm just skipping this slide. Okay, deoxyribose and pentose level phosphate groups, and in organic bases, these are the adenine, guanine, cytosine, and thymine. And here these are regarded as purine and pyrimidine bases. And in purine bases, actually they are, these are the configuration of the DNA and RNA. So I am skipping this purine bases description. Okay, students. And nucleosides, you know, after the what happens in nucleosides, that these are getting after the deletion or extraction of the phosphate groups from the nucleic acid chemical composition. See here, here the ribonucleic acid in ribonucleotides. So what are the side nucleosides and the nucleotides? In nucleosides, adenine, cytidine, guanosine, and uridine. Here these are the, in case of the ribonucleotides, adenyllic acid. Here adenosine, it is adenyllic acid. So these are the acidic forms. Okay, and in DNA level, just like that, these are also in acidic forms. Deoxyadenosine, this is deoxyadenyllic acid. These are the coding, you have to remember it. The primary structure of the, this, I am also skipping this structure because you have already known from your basic high school level. Okay, as I have a limited time, and you know, DNA double helix was Watson and Crick model during 1953, and for that he those persons, Watson and Crick got Nobel Prize. Okay, and the Chargaff rules, chemical analysis of Chargaff rules, the number of purines and the number of pyrimidines are equal in number. In DNA content, A is equal to quantity. So, otherwise, there may not be the configuration or the attachment of the adenosine with the thymine. So, basically, the purines and the number of pyrimidines, they must be equal for the double helix and double hydrogen bond and triple hydrogen bond. And in triple hydrogen bond, G combines with the C. Okay, and these are the features of the DNA double helix structure. Just I told you what A combines with T with a double hydrogen bond, and C combines with G with the triple hydrogen bond. Okay, this is the configuration of the DNA double helix structure, and a helix in a DNA structure that is 20 angstrom in diameter. And these are the pairing complementary fashion where hydrogen bond is double, where hydrogen bond is triple. So these are the points. Okay, actually in a term in a 10 base pairs are present in a loop formation. Double helix form so many loops are there. So in a particular loop 10 base pairs are found in a normal configuration, and in unit 12, that is the genetics of bacteria and bacteria phages. Okay, so in bacteriophages, that is the virus that can infect bacteria, referred to as bacterial parasites, and each phage may infect a single or multiple bacteria depending on the entry of the bacteriophage. And that is, see here, bacteriophage structure, head and the tail portion, and in between the head and tail portion there is the neck region, and this is the contractile sheath of the tail region, and there is a base plate with the tail fibers. So this is the structure of a ultrastructure of a bacteriophage here. Okay, uh, and then bacteriophage genome genetic materials are ssRNA, dsRNA, ssDNA, or dsDNA. Okay, and its life cycle, see here, uh, it is the lytic cycle, attachment, penetration, biosynthesis, maturation, and release. Okay. And the lysogenic cycle, attachment, penetration, prophase, cell division, biosynthesis. And in genetic configuration or transformation, genetic recombination, in which DNA fragments from a dead degraded bacterium enters a competent recipient bacterium, and it is exchanged for a piece of the recipient's DNA. It involves 4 steps. Now, that is, one is transduction. Okay, and another is bacterial conjugation, uh, and third one is A plus conjugation process, and another one is hyperconjugation. Okay, and in the resistant plasmid conjugation, genetic recombination, in which there is a transfer of R plasmid. And what is R plasmid? That R plasmid, a plasmid coding for multiple antibiotic resistant and upon a 6 pilus. Okay. Okay, thank you, students.
Thank you, ma'am.
Hello.
Thank you, ma'am.
Okay, thank
Thank
you.
you, ma'am.
We should leave now. Thank you. Thank you.